Hello everyone, today we're going to talk about uniconts, an introduction to this taxonomic group. We're going to start by looking at amoebozoa, and in the uniconts it includes both these two taxa, the amoebozoa and the opisthocons. So we're talking about eukaryotic organisms, and the amoebozoa are going to be single-celled, whereas in the opisthocons we can have both single-celled or multi-celled organisms. So the uniconts are characterized by having some genetic modifications compared to all the other taxa. So they've had a rearrangement of chromosomes compared to these other groups. So that's a specific genetic difference that we can see in the uniconts. So amoebozoa and our opisthocons have this uniconts characteristic genome, and then the opisthocons have a characteristic single flagellum if they do have one. So that flagellum is the special characteristic of the opisthocons. And if we zoom in on the opisthocons, we see that we have these two major clades, the amoebozoa. These are single-celled organisms. There are sort of two groups that are these amoeba-like organisms, and then there's this other weird group called the slime molds. Now, these are really fascinating organisms. We aren't going to get into those in our lab, but hopefully you'll have a chance to learn some more about them. They have strange qualities. They live most of their lives as single cells, but they can also swarm together and form a crawling slug. And they have similarities to fungi in the places you find them and in their general appearance, although they don't grow a mushroom. They grow sort of a network across the surface. So the tubulinids is, excuse me, one of the taxa of amoebozoa, the most common kind that we find out in the environment. That's not disease-causing organisms, although some are. And we'll focus on just one species, amoeba, the typical lab organism. Now within the opisthocons, there's two major clades. There's one the fungi are in. The closest relative of the fungi here in the nuclearids is actually chytrid. They're single-celled organisms in the nuclearids. So we have a single-celled organism that is the closest relative to fungi, which can be multi-celled. We also see in the animals, we have these choanoflagellates that are single-celled that are the closest relative to the animals. So we have the ability to be multi-celled as an apomorphy that arose dependently in these two lineages. Now we're going to spend the rest of the semester on animals, so don't get excited about talking about them today. We're going to do the amoeba and fungi, and then we'll shift gears to animals for the rest of the semester. Fungi are a very old lineage. They came into the terrestrial environment along with plants, possibly before them, and they exist in aquatic environments as well. There's about 10,000 species, and some individuals can be very, very old, over 1,000 years. They can be very, very large, the size of multiple football fields. So they're a very interesting group. The more I have learned about them, the more interesting I find them. All right, real quickly, our amoeba is a single-celled eukaryote that's going to be characterized by being heterotrophic, and it's going to use its pseudopods both for locomotion and for feeding. So it has these large, thick pseudopods. And this irregular shape that characterizes the amoeba is different than any other single-celled organism that we're going to see. If you're watching one, you can see that its outline changes shape from one few minutes to the next. And if we see different individuals, then they're going to look slightly different from one another. As far as cells go, they're relatively large. Some of the largest single-celled organisms are deep-sea amoeba. You can see from this scale bar that they're 1/10 of a millimeter. They're almost visible to the naked eye, which is really big for a single-celled organism. And they, as you might guess from having this very flexible outline is that they have no cell wall. So this is a couple different ways of viewing amoeba. We have a video of live specimens. You can watch them actually crawling. You can see the extensions of their pseudopods and then how they drag themselves along using that. All right, that's it for amoeba. Now we're going to spend the rest of the lab talking about diversity of fungi and the characteristics of the different groups. So there's 3 different groups of within the fungi, and when we think about what they are, we often think about different kinds of edible mushrooms. So depending on where you live, different species of fungi grow and are used in cooking, but almost every culture has some kind of delicacy of different kinds of fungus, ranging from shiitake and enoki mushrooms in Asia to morel and truffle mushrooms in Europe, and in Central America, huitlacoche, which is the corn fungus that it's a type of mushroom that grows on corn. So they can be very important in agricultural sense and in different products that we use that can be also made from mushroom fiber, sort of. All right, the multi-celled organisms that we see here may or may not have a single-celled part of their life cycle. Some species of fungi only occur as yeasts, which would be single-celled organisms. We see these colonies like we've already seen on our settling plates with the auger plates that we used earlier in the semester. If we zoom in and look at a single cell of yeast, they're just sort of a roundish shaped cell. We see that they're going to divide here, they're going to bud off a new individual. So those colonies can grow quite rapidly. The other colonies of organisms that we might find on our settling plates, we already talked about other kinds of molds and things like these big fuzzy molds. So we'll see today that those molds are actually the structures that are used for asexual reproduction or sexual reproduction, and the little fibers that are sticking up have a ball on the top, and spores are going to come out of those balls. So the ecological role of fungi is as decomposers, and the way that they're going to go about doing that is that the bulk of the organism is going to be down here in the soil. And so the cells that make up the body are called hyphae, and the whole body of the organism is called a mycelium. And they're heterotrophs, so they're going to be secreting digestive enzymes that are going to come out of the cells into the soil around there. And any kind of organic matter, that is dead plants, dead animals, other kinds of broken down, previously living material, it's going to get digested and broken apart, and they're going to be able to then absorb the molecules of food that are breaking off of those slightly larger structures. So they're going to be absorbing nutrient molecules from the soil around them because they have digested it from outside of their body. The structure that we see that's growing above the ground, in this case the mushroom, is a reproductive structure. And depending on the taxa, it could be asexual or sexual. And often these are referred to as fruiting bodies. So they are the reproductive structure sticking up above the ground so that it can spread those spores across a larger area. All right, when we look at where in the cycle of the life does this fruiting body grow from? So when we think about the fruit that comes onto a plant, a flowering plant, it grows the flower, and then if that gets fertilized, then the fruit is going to grow at the base of the flower, and then the seeds are going to be in that fruit, and that seed has the embryo inside of it. It's the next generation. So when we look at where in the life cycle of the fungus these fruiting bodies occur, we have to look at some weird stuff that fungi do. So most of them do asexual reproduction, but not all. So one thing that can happen if we have an existing mycelium, right, is that it can grow these spore-producing structures, and a spore-producing structure is called a sporangia, just like it is in plants. A spore-producing structure is a little stalk with a ball on top of it. The spores are going to come out, and those spores are going to grow into a new mycelium. Now, if we look onto this figure, we see that we're in the haploid part of the life cycle. So in the mushroom life cycle, they only have that multi-celled haploid organism. It's the opposite of animals, and it's only half of what plants do, remember. Right? So this is going to be a haploid individual. Right, now, when we have two different haploid individuals that are growing near one another, and they are of opposite mating types. Now in fungi, we don't have any characteristics that we can use to identify individuals as male and female, but they do have mating types, and they work the same way. They have to be of the opposite mating type in order for them to be able to have, to do sexual reproduction. So sexual reproduction in animals is going to occur through the fusion of gametes, and we have the sperm and we have the egg. So there's an equivalent to that that happens in fungi, and this is in more detail than we've talked about before. Right? And we're going to have these two different individuals. I'm going to indicate what happens to the nucleus. I'm going to say one of them, the negative, has a white nucleus, the plus has a black. So these are two hyphae that are coming towards one another in the soil, and they say, aha, you are the opposite mating type, and wait, what am I doing? You are the opposite mating type, and I want to fuse with you. All right, so fertilization is the fusion of gametes when we're talking about animals and plants. Something a little different happens here in fungi. So the first step is this plasmogamy, right? Plasmogamy is going to be the fusion of the cytoplasm without the fusion of the nuclei. So what does this look like? We have these two cells that come together and they fuse. So there's a partition or a barrier that separates them from the cytoplasm of the rest of the individual, right? And we're going to have the two nuclei, but the nuclei aren't going to fuse with one another. So we just have fusion of cytoplasm. We don't have fusion of nuclei. In animal and plant reproduction, the fusion of the nuclei happens immediately, right? So there isn't a separate step where this happens. Now, in fungi, this can exist for a very long period of time and nothing else happens. So they fuse together. The nuclei don't fuse though, right? So we have a different name. This cell isn't diploid, this cell isn't haploid. We say that it's heterokaryotic rather than either being diploid or haploid because it has two separate sets of chromosomes that aren't working together. They have not been bundled together into one nucleus. So plasmogamy is going to lead to this heterokaryotic stage. So at some point later on, when conditions are good, this cell that's in the heterokaryotic stage is going to be the thing that grows up into the mushroom. Right? And so all of the cells that are in the mushroom grew from those two the cell that had those two separate nuclei in it. All right, so all of the cells that are in that sexual reproductive structure are heterokaryotic. Now, somewhere inside the body of that fruiting structure or reproductive structure is going to be a place where the next stage is going to happen, right? So this happens in a special location in the body of this organism. And what's going to happen is that finally we have the fusion of the nuclei, all right? And the name of the step that causes that to happen is called karyogamy. And what we end up happening is that this zygote is formed. And we finally have those two different contributions of genetic information fused together into a diploid state. Now, the zygote only exists for a very brief period of time. It almost immediately goes through meiosis and it makes these sexual spores. So these are the product of sexual reproduction. So we do have spores that come from sexual reproduction. We also have spores over here that come from asexual reproduction. Their structure is slightly different. So these spores that come from asexual reproduction are not as heavily covered with the protective molecule coating, so they don't last as long, they aren't able to survive as well. So the spores from the sexual reproduction, there may be fewer of them, but they can survive better over a longer time in the environment. Right now, each of these spores is going to go off on its own somewhere, and it's going to start to grow up into a new individual that's genetically distinct from the parent. All right, we have 3 different taxonomic groups within the fungi: the Zygomycota, the Basidiomycota, and the Ascomycota. So notice the mycota part of those words is all the same. So basically we have these 3 different taxonomic groups. You just need to remember them as A, B, or Z. Right? So for A, B, and Z, we're starting with Z in reverse order. And we're going to look at each of these 3 taxa. We're going to look at their sexual reproductive structure and their asexual reproductive structure if they have one. Right, so we've looked at these sporangia under a dissecting microscope, and you're going to be able to see that the spores are coming out of them. Here you can see spores coming out. So if we were to zoom in on this bread mold, what we actually would see, right, these visible to the naked eye structures, there are enough of them, we can see them with the naked eye. These are going to be the sporangia, right? So they make spores, that's all we're going to call them. So for Zygomycota, we're just going to call their asexual reproductive structure sporangia. Now, somewhere deep down inside this bread, right, way deep down inside, even if you pick off the furry part, you're probably going to still have some of this fungi on the inside. This is the place where those tips of the hyphae have fused together. Can you see we have these 2 different hyphae here? And this ball that's formed in the middle is that heterokaryotic structure that is going to end up releasing the spores that are coming from sexual reproduction. So this is microscopic. This is not visible to the naked eye. In the Zygomycota, you don't see a mushroom-like structure that's going to grow up and be something you can see out in the environment. We're only going to see these on a microscope preparation. So to distinguish these from the regular old sporangia, these sexual reproductive structures are going to be called zygosporangia. So these are the sexual reproductive structures, and the sporangia down here are asexual. All right. Next group is the Ascomycota, and these are sometimes called cup fungus. All right, notice this finger here, and these fungi are characterized by, instead of having a mushroom that looks like a right side up umbrella, right, one that's protecting you from the rain, the cup fungi looks like an upside down umbrella, right? It's more of a cup shaped rather than a dome shape. And they tend to be quite small, right? So if you see that you have a concave structure, it's tiny, it might be among some moss like it is in that picture, it might be on the forest floor. The person who's taking the picture might put something in the picture to help you tell that it's a very small individual. If somebody's holding up a mushroom that's, you know, big enough for you to see, that's bigger than their hand or as big as their hand, it's never going to be Ascomycota. These are very petite organisms. So this structure that sticks up above the ground, this cup-like structure, is going to be called the ascocarp. And this is the structure that has grown up from those 2 fused heterokaryotic cells. So the cells that are inside this cup structure are heterokaryotic, except for the place where there is going to be the process of karyogamy and then meiosis. And the place where that happens is in structures. So remember, we have meiosis happen inside the body of animals in special body organs called testes or ovaries. Inside the body of the fungus, inside the ascocarp, there is a body organ. It's very small, and there's many of them that are going to be the place where that fusion of the nuclei occurs. So we end up with the single-celled, truly diploid zygote that immediately divides into 8 spores, right? So we're going to find these products of sexual reproduction in the Ascomycota. We're going to call the structure that where that meiosis takes place, we're going to call it an ascus. There's only one, it's an ascus. If there's multiple, where they're acini, right? Not acai like the berries, but acini. So we're going to see these spores that are inside these structures. They're sort of oblong, long and thin. There's bunches of them. So the inside surface of our cup fungus is going to be the tips of all of these acini coming together, right? So if we are looking at the inside, we're seeing the tips of these little structures. All right. So that is the sexual reproductive structure. And so that's the ascocarp, right? That is visible to the naked eye. Now the acini are not visible to the naked eye. We're going to have to see those in a microscope slide. Does the Ascomycota have a asexual reproductive structure? Yes, it does, right? There's a lot of variation, but basically these are the same kinds of things as sporangia, except instead of sporangia, they are called conidiophores, right? The individual spores are called conidia, but notice how instead of there being a ball that's going to release the spores out of it, like we saw in the Zygomycota, that we have these spores that are kind of sitting on the top of these structures, and they grow up in a pile and then they start to fall off. All right, so I like to think of these conidia as being a sporangia that's having a bad hair day. Instead of having all the spores inside of it, they're stacked up on top. When we look at different taxa, when we look at different genera and different species, one of the things that looks different is the shape and the structure and how many spores they build. So these are just some different examples. There's quite a lot of diversity in the conidia. So I will only show you ask you to identify pictures that are very similar to the ones that you've seen here, because there's just so much diversity in it. All right, so these are the asexual reproductive structures of the Ascomycota. They're called conidiophores. All right, our last taxonomic grouping within the fungi is called the Basidiomycota. Now these are nice because these are the familiar mushrooms that you already know about, and they do sexual reproduction with a structure that is the mushroom, but its fancy scientific technical name is a basidiocarp. Now remember that that multi-celled heterokaryotic structure in the Ascomycota was called the ascocarp, and here we are, the Basidiomycota, it's called the basidiocarp, right? And in the ovary of the flower, the place where the reproductive structures are, the place where the egg is going to get formed, is called the carpel, right? So that word is telling us that that's where reproduction is going to be happening. So if we look at our mushroom and we see that there's these gills up underneath it, and we zoom in on the gills, so each of these is one of these little gills that's hanging down, and we look at the surface of it, we'll see that we have a place where the spores are being made. Right? So the spores are going to be produced on this structure. It's called a basidium. Right? So the basidium is the equivalent of the ascus. The basidium is going to be equivalent to the testes or the ovaries, which is where the meiosis is going to happen, and we're going to produce those spores. So we have several different microscopic views of these different structures for you to look at in lab today. And our last group that we're going to talk about is a really interesting group of organisms called lichen. And often we find lichen on rocks. And so this sort of green crud that's sticking on this rock saying, I'm really liking you, is something a student sent me in order to help me help you remember that lichen is pronounced lichen. It's not lichen, it's not something else, it's lichen, as in, I like you. All right, so we find lichen on rocks, we find it on concrete, we find it on living wood sometimes, we find it on other structures, but it's not getting its nutrition from them. So it's going to be sitting on top. And the reason it can do that is because it requires very, very few nutrients. And in fact, the lichen is going to be an organism that we find doing what's called primary succession. So primary succession is going to be basically an organism that colonizes new habitat. But in the case of primary succession, it isn't habitat that's, say, been burnt by a fire where there were living things previously. Primary succession is the first organism that's going to come in and it's going to be able to live in a place where there is no soil, there are no nutrients, there are no other organisms living there. So if you think about a volcano erupting and building land out in the middle of the ocean and forming volcanic rock, some little spores are going to drift in on the wind of lichen. It's going to land on there and it's going to start growing. It doesn't need anything else in order to grow. And it's actually going to start secreting some acids from its body, and it's going to start degrading that rock and actually turning it into soil. So it's an organism that's very important in the history of life on the planet. It has helped turn these barren new environments into environments that other organisms can live in. All right, what is lichen? Well, it's a little hard to understand exactly what it is because it is a mutualism. And that means that it's going to have at least two different species that are integrated together in such a way that they're going to be sharing resources and helping one another. So we are going to have a fungus. That's the reason we're talking about lichen today, is because it's going to be some kind of fungus, usually an Ascomycota, and an algae or a cyanobacteria. So we have the fungus that's going to provide some physical structure for the organism. And then we have the algae or the cyanobacteria being held inside the structure of its body and doing photosynthesis, right? So these organisms need to get a very tiny amount of additional nutrients that basically is going to fall onto them due to rain or dust. But other than that, they are going to be waterproof, sitting there doing photosynthesis, and able to live in environments that almost no other organism does. When we look at diversity in lichens, there's a great number of species that it can be very hard to tell apart, but when we look at how they grow, that's one way that we can start to think about what's happening for this sort of body or the structure of the lichen. So they can either grow in a flat, thin sheet, which is really to form, they form a crust, which is just going to grow across the surface of the rock. So those are crustose, right? They form a crust. Fruticose is they're going to stick up almost like a tree, like a branching structure that's going to have these spindly stalks that are going to stick up. So that's a fruticose structure. And then if we have ones that look leafy, they have more, they're coming up off the surface of the rock in these thin sheets. We're going to say they are foliose. Oops. Foliose, like foliage. All right. Now notice the colors we have here. They can be a relatively bright green. That color is due to the photosynthetic pigments of the algae that's living in them, but they can also be black or gray or yellow or red or orange. But then they also sort of look dry, and they can be this sort of dusty green looking. I'm partial to these lime green ones. I like those guys a lot. All right, let's look at the inside structure of the lichen. Oh, one more thing is if they are capable of asexual and sexual reproduction. But basically the organisms that are part of this association each do their own reproduction. So the fungus that's going to do its reproduction, it's going to grow an ascocarp. So sometimes we'll see that we have on our lichen, it'll have those little cup-like structures growing up from it. Okay, if we take a cross-section through, we'll see that there's sort of some acellular stuff on the surface of the lichen. And then this structure at the very surface that's stained darkly is very waterproof. It's built out of that chitin molecule that makes up the cell wall of the fungi. And so it has this tough protective surface. And we go to the bottom of it, it also has a very tough protective coating. It might have some little extensions sticking out that help anchor it onto the rock. The space inside of the body structure has some air spaces, right? These white spaces are actually empty spaces, and that's going to allow carbon dioxide and oxygen to move throughout the structure, and it's going to be able to get the carbon dioxide to these algal cells. So these little circular cells are the algae that are being held inside the body of the fungus. Now notice they're being held towards the top. We're going to have sunlight coming in. We want them to be able to get to those cells. And so we see that these algal cells are actually embedded into the overall three-dimensional structure of the lichen. Right? Once you start looking for lichen outside, you're going to be able to notice it in a lot of different places. San Diego isn't an amazing place for lichen, but you will see it. You will see it around, especially if you're in an area that's a little bit shadier. Say if you go down into a canyon behind campus, you'll find it growing on the branches of the shrubs that are down there. All right, that is the end of our lecture on amoebozoa and the opisthecons, but just fungi. We'll start with animals next week.